All right, thank you very much for having me. Wonderful. So my talk's gonna be a little bit different. I'm going to talk about a technology that relates very much to significance of hybrid capture approaches, and that's why I'm talking here today. We're a company called TwinStrand, which is a spin-off of the University of Washington, and we have a technology called duplex sequencing. And duplex sequencing is basically a technique for improving sequencing accuracy to a very high degree. So with standard Illumina sequencing, there's a challenge where low frequency variants are somewhat difficult to detect. If one is looking for a difference between a tumor and a normal, or between one patient and the other where most of the cells have the same genetic variation, it's relatively easy to do. The signal to noise ratio is quite high. On the other hand, when there's very low frequency variants, like one cancer cell with one mutation mixed in with 10,000 normal cells, the error rate of standard sequencing becomes limiting, and it makes it hard to see those variants. So just to put this in perspective, this is a representative 3,000 base pair portion of a genome. A heterozygous or homozygous mutation will show up as 50% or 100% roughly of the reads in that sample. But if one zooms into the low frequency variants right down near the bottom here, you see a little bit of haze, and that becomes pretty significant zoomed into that magnification. You can see every position in this region has a mutation somewhere around 1% of the reads, meaning that if you had a true mutation present at 1 in 1,000, it would be really hard to tell that from the noise. So what duplex sequencing is this is a double-strand tag-based error correction technique that lets us take standard Illumina sequencing, do special biochemistry at the front, put it in a routine Illumina or other sequencer, and do special informatics in the back that improve accuracy around 100,000-fold. So the analogy is a low sensitivity digital camera sensor versus a high one. It lets you see very faint details. The basic idea how it works is DNA is a double-stranded molecule. So in nature, one half of a double helix reciprocally encodes the other half. And so for much of life on Earth, the way replication fidelity is maintained between cell divisions is that there's an error correction comparing one half of the molecule to the other after cells divide, and a mismatch is recognized as an error that needs to be fixed. We recapitulate that solution biochemically. So the way duplex sequencing works is we have a unique label that's affixed to every single molecule. So in this particular case, those N's represent a random degenerate sequence. In this particular version, the top sequence and the bottom sequence, there's N and N primed. That just means there's a complementary randomized sequence. In this case, there's 12 N's, which means there's 4 to the 12 possible combinations. If we ligate one of these adapters in one end and one in the other end, we have 4 to the 24th possible random combinations, and there's just simply no way that two molecules are going to have the same label by random chance. So here I'm showing random sequence alpha and random sequence beta, just two arbitrary random sequences on the different ends of the molecule. And what you can see is both the top strand and the bottom strand have that same random sequence in them, but in the top strand, random sequence alpha is next to primer site 1, and in the bottom sequence, it's next to primer site 2. So there's an informatic way to relate those strands, but also distinguish them, and those are the fundamental aspects of duplex sequencing. So looking at one particular molecule among a population of many that are being sequenced with a particular tag on it, I've simplified it to show just the top strand conformation or bottom strand conformation of that particular tag as purple or green. AT represents a true mutation in this particular molecule. In the second column, so what we do, we ligate these adapters, we do amplification, then we do hybrid capture enrichment, which I'll talk about more in a minute, and then sequencing. And then we look in the tag region and we say everything that has the same tag in either the top strand conformation or the bottom strand conformation gets grouped together because those things all must have necessarily come from the same starting molecule. Any variation among the actual sequences themselves we know must be an error because everything came from the same starting molecule. So we can average those out and say, well, the only thing that's likely to be real here is this A. All these red bases are errors from PCR sequencing or cluster amplification. We do that for the top strand and we can do that for the bottom strand. One thing that actually happens not infrequently, you'll see here there's a black T present, all these molecules in a red C. This red C is an error, and it's an error that happened during the first round of PCR. It's a jackpot event that occurred and was propagated to all derivative copies, and this happens at sites of 8-oxyguanine and deaminated cytosine and 5-methylcytosine in particular. So when you create a consensus of those, in the third column, you'll see that C gets carried forward. If you weren't able to distinguish the top and bottom strands, you wouldn't know if that C was real or was not. And so what duplex sequencing lets us do is take that UMI approach and then truly compare the two strands of the original duplex molecule together, and we see there's not a corresponding mutation on the other side, and we get to an accurate representation of the molecule. Error rate of the standard sequencing between 1 in 100, 1 in 1,000. Third column, single-stranded UMI technology is 1 in 10,000 to 1 in 50,000 or so, and duplex sequencing is below 1 in 10 million. So I just showed you this figure a minute ago. Standard sequencing, duplex sequencing, the exact same data. What you see on the bottom is that all the noise goes away and there's only one true mutation present in this sample. This particular sample is a patient with chronic myeloid leukemia who's getting a particular targeted drug. This patient relapsed 4.5 months later, but after the sample was taken. And I, as an oncologist, had I known that mutation was there at an increasing frequency, would have said, well, there's 4 other FDA-approved drugs in this space, and several of them are not cross-resistant to that mutation. I could have switched therapy and it prevented this relapse. And with targeted drugs in oncology, evolution of resistance is a ubiquitous problem. The more and more new drugs we have, the more frequently find this problem. So that's just one example of how something's actionable that's necessary to look for low-frequency variants. So talk briefly about hybrid capture. Duplex sequencing cannot be done with amplicon methods because you have to ligate a duplex tag to both strands. So you can't, a primer with a tag simply won't work. There's no way to relate the two strands together. So we necessarily always work with hybrid capture approaches, and the efficiency of those is really critical for the efficiency of our approach. So after one PCR cycle, we have 2 amplicons that are generated, one that came from the top strand, one that came from the bottom strand. They both have alpha beta, but alpha is next to primer site 1 in the top, and it's next to primer site 2 in the bottom. Then after we make multiple copies, we denature them and we do hybrid capture. People always ask me, well, how do you do hybrid capture on and recover both strands? Wouldn't the probes stick together? The answer is that you make copies of each, so you get a copy of the top strand and the bottom strand in both the sense and anti-sense orientation. For duplex sequencing to work, we want to recover copies of both strands. We want to recover multiple copies of both strands, and we want to do it in an even way. We don't want to recover 10,000 copies of one strand and 1 copy of one. It becomes very inefficient and expensive. And so having a really robust hybrid capture is critical for us keeping costs down to be able to look at this level of accuracy. So I'm going to show you a couple other case studies. The first thing I want to say here is that duplex sequencing is a tool, and it's a really good tool, but for certain applications where you're looking for genetic difference between, say, person A and person B, it's really excessive. You don't need that level of accuracy. But applications like I just showed, where you're looking for low-frequency variants, it's really critical to get rid of that noise. So 4 case studies. The first one I already just showed you about, targeted drug resistance in oncology. Second one is an interesting one. We have a substantial project with several clinical groups in US and Europe developing a method for early detection of ovarian cancer. So the reason we do mammograms, pap smears, and colonoscopies for screening for other cancers is the idea that the earlier you detect the cancer, the more treatable it is. Ovarian cancer is unfortunately, like pancreatic cancer, typically detected very late once it's already spread. And if we could simply shift the time of detection to when something was stage 1, the cure rate would be very high, above 95%. There is no current test that's ever been proven to really work in this disease. So our approach here is interesting. We work with a collaborating group that's developed this fairly simple catheter device that during a pap smear, which is a routine cervical cancer screening test, you advance this tiny little, very narrow tube, and I know that's very easy for me to say as a male, but it is relatively benign addition to a routine pap smear. It takes about 2 minutes. You flush in 10 milliliters of saline, you draw that back out, and the idea is that very early ovarian cancers slough off small numbers of cells that float down the fallopian tubes and into the uterus, and the idea is that we can collect those cell debris, deep sequence it for a gene that's mutated in essentially all high-grade serous ovarian cancers, and look at that low frequency variants as a marker of an early cancer is the concept. Similar type of figure to what I showed you a minute ago. This is standard sequencing. Every spot on the surface corresponds to one position in P53, which is a gene mutated in essentially all high-grade serous ovarian cancers. duplex sequencing of the exact same data. This peak in the back, 1 in 1,000, is a mutation that came from a stage 1 ovarian cancer patient. This woman showed up in the emergency room and got a CAT scan for kidney stones, and incidentally, a small mass was detected. She was enrolled in a clinical study. Prior to a diagnostic surgery, a lavage was done. We identified this, and lo and behold, that turned out to be the same mutation that was found in the surgical sample. These 3 mutations in the foreground at much lower frequencies, those are not errors. That's the residual biological background of normal human aging. In this larger study, which we have up in bioarchives right now, if we ignore the tumor mutations, we can predict roughly how old the women were in the study based on their background to these low-frequency mutations. And if we know how old they are, I think we can predict if they're a smoker or not. We can see modulation of this based on genotoxic exposures, which is pretty cool. Okay, third case study. This is something you've potentially seen, a variety of other folks working on, is the concept of residual disease and acute myeloid leukemia. One of the best predictors of relapse in acute myeloid leukemia, which is sort of a fairly aggressive, predominantly adult leukemia, is after we do this big whopping slug of multi-drug chemotherapy, and you take a sample of blood or bone marrow, if you can detect some residual leukemia cells left, that's sort of not a great sign. It means you probably need more treatment, or you should go into a clinical trial of a new drug. And maybe if it's all cleared, your risk of relapse is lower. Maybe we can spare you therapy or bone marrow transplant, some other very highly morbid treatments. Challenge of this is how do we do this? The flow cytometry is the kind of current gold standard for this detection, and it's really good in some centers, less good in other centers, and it's pretty variable between patients, partly because it relies on the immunophenotype of the leukemia cells. Some are very distinct from normal, some are very similar. So a concept of how we could do this better is to say, well, we know leukemias are driven by mutations, so if we identify those mutations at baseline, and then we look for them at low frequency after treatment, and we see that they're still there, that's a way that we could potentially do this. The problem is the error rate of standard sequencing. This is standard sequencing. This is using NextSeq, which is a new generation chemistry. You can see every position in this. This is roughly 600 base pair chunk of the genome that we're looking at, representative one. Looks like it's mutated at least 1 in 1,000. This is a single strand tag-based error correction, something that many different folks have used, and you'll see this referred to as UMI correction. That drops error rate considerably, but you see that there is still quite a bit left. And the bottom, duplex sequencing, essentially removes all of the errors. This experiment was actually taking normal peripheral blood DNA from a healthy young individual, spiking at AML patient samples or cell lines that have specific driver mutations at levels from 1 in 100 to 1 in 100,000. We sequenced a 1 million-fold duplex depth. Can't see any of the variants here. You can see down to about 1 in 1,000 here. Here we have no problem seeing down to 1 in 100,000, which is significantly higher than anything else available. Last case I'm going to show you is something very much outside the field of clinical oncology. It's an area called genotoxicity, which is a field that is rooted in kind of fairly old techniques. This is the idea that every drug or chemical that goes into humans needs to be tested for whether it's a carcinogen. And the current standard way you do that, you take a bunch of rats, half of them you give the chemical to, half you don't. You wait a couple of years and you cut them up in little pieces and count the number of tumors. And it's pretty expensive, it's pretty slow, it's kind of medieval. And if you're a drug company that's put a lot of money into making a drug that potentially could really help people, you're twiddling your thumbs for 2 or 3 years waiting for results. Theoretically, if you just directly sequence normal healthy tissue after exposure to these chemicals, we could get that information much more quickly. The problem is the background mutation rate of normal healthy tissue is about 1 in 10 million. So the error rate of standard sequencing is way too high for this to work. This is doing duplex sequencing, looking at mutation frequency. The green group, each one of these bars is a replicate animal. This is an untreated group. The second group is treated with benzoate-pyrene, which is a major component of cigarette smoke. It's a mutagen. And the third is ENU. It's an alkylating agent. And from across the room, you can say, well, whoop-de-doo, orange, blue, those are much higher than green. Anybody can see that. Well, the point is, look at the y-axis. We're looking at the difference between 1 in a million and 2 per 10 million. So remember what I said about the magnitude of background. So on this screen, this last time I gave this, it was on a much bigger screen. So pretend that that's 5 feet. If you extrapolate that 5 feet difference to the background error rate of normal Illumina sequencing, the background error rate would be literally 50,000 feet above our head. There's no way you could see that difference without this level of error correction. It is possible to not only look at mutation frequency, we can look at spectrum, we can look at patterns, we can infer mechanism based on the particular types of mutations, whether it's an A to G or T to C. We can cluster them. Just a computer, without knowing anything different, can group these mutagens in treated animals into different groups. And again, these are mutations that are present levels of like 1 in a million. Some of you have seen this type of data. This is trinucleotide mutation signature data. It's basically you divide up every mutation, not just by whether it's a C to A or T to C, but by the adjacent nucleotides, whether it's an A between two C's or between a G and an A, something like that. And each one of these comes up with a very specific pattern. So benzoate gives a very specific pattern, ENU gives a specific pattern, urethane gives a specific pattern. And urethane, you see this in benzoate, there's almost no T to A's. And urethane, you see this spike, spike, spike. That's at a motif NTG, which for whatever reason is very mutation prone. So that's pretty cool. And these are mutations, again, at levels of 1 in a million. This is benzoate in healthy tissue of mice a couple weeks after exposure. This is taking all data in the entirety of the TCGA database of smoking-related lung cancers. So these are humans that have been exposed to benzoate and other mutagens for decades and ultimately developed a tumor that cloned that founding cell, and the exome mutations or whole genome mutations show up there. We see the same exact thing a couple weeks after treating mice in normal healthy tissue and recapitulating exactly what goes on in that situation. And the only way we can do this is because we can see 1 in 10 million. So a lot of different applications. And I'm not going to talk about any more of those, but that's just to give you a bit of a flavor of what we can do. Fundamentally, this is all about a genetic needle in a haystack problem. So duplex sequencing, getting into a little more of the nitty gritty in the hybrid capture, which is important. So we have to do hybrid capture. And to look at very low frequency events, say, for example, in AML cases, we typically have to sequence to pretty high depths, right? You can't see 1 in 100,000 if you only have 100 copies that you're sequencing. So typically, the cost of duplex sequencing reflects how wide the panel is, how many kilobases or megabases, how deep you go, whether you're looking at 10x depth or 100,000x depth, and the duplication factor, because we're deliberately making copies of both strands. So for sort of really high duplex depths, where we look for very low frequency events, we typically try to stay between 1 kb and 100 kb, because beyond that, the costs do go up. For sort of modest depth sequencing, we're looking at 5,000, 10,000. You can go up to a megabase pretty reasonably. So for us, the panels we design range from as little as 1,000 or 500 bases up to megabase. And so that's a pretty big range of panels. A lot of people are working with kind of standard size panels of 50 kb or 100 kb, 200. It's pretty unusual to go to these really small panels, but we actually need to be able to work with all of those. So there's different approaches for doing hybrid capture. On one extreme is ordering individual oligos. So you basically order an individual oligo. It's 100, 120 mer with a biotin on it. It comes with an individual tube. You mix them together to make up a panel. On the other end of the extreme is array technology, where you're ordering an array of large numbers of oligos that are on a surface. They tend to be not as accurately synthesized. It tends to be really mostly set up for very large panels. And then there's Twist, which is this kind of newer player, I guess not that new, but it really fits in this kind of nice middle category, whereby with individual oligos, these work fine for small panels, but if you're sort of making larger panels, they get very expensive, and it takes a fair amount of time to do this sort of manual adjustment yourself, or order these large panels, simply because standard approaches for individual oligos, there's a lot of mass spec QC. With really big arrays, that's similar. It's kind of difficult to get those panels going, and so we found that a lot of things we do, we need really quick turnaround. We need to be able to work from 1 kb up to a megabase, and we want to be able to do this at sort of reasonable costs. And so Twist is something we started using a bit this year. I've been really pleased with. I'm going to show you some comparison data. One of the strengths of this approach is that you simply give an interval file, and they use kind of some software and experience from ordering, working with different sequences to kind of automatically balance and tile across particular regions. And I was, pretty skeptical of this concept, but we'll show you what actually happened. And then for the larger panels, it just is a lot more cost effective. But the most important thing for us is because we go through so many of these things and need to do rapid prototyping, it only takes a few weeks for these to come back around. So it's a really nice kind of middle of the road approach that we've been using a bit more now. So there we go. One of the things, we did a couple recent comparison experiments where we took this AML residual disease panel, which is about 50 kb, and a sub-panel, it was about 1 kb, and I said, all right, you guys, you want to prove that you're as good as us ordering individual oligos and manually going through and jiggling and reordering and testing? No way. So I just gave them the interval files, and then 3 weeks later, this twist panel showed up. And what we did is we did a comparison. We used a relatively small amount of DNA to start because I didn't necessarily believe this was actually going to work. We sheared it, we ligated with duplex adapters, we generated our duplex library, and we did hybrid capture comparing our several-fold optimized individual oligo panel and this off-the-shelf twist panel, or automatically designed twist panel. And what we saw was pretty surprising that for both the larger panel and the smaller panel, typically smaller panels don't perform as well in our experience without optimization because the enrichment requirement is higher, perform quite well, about 90% on target rate for both. We picked this panel in particular because it's sort of complex. There's a lot of repetitive elements and pseudogenes and things that are kind of hard to work around. And what we saw, is very, very similar for both the large panel and small panel. It was like, 1 or 2% different, lower in the large panel and 1 or 2% higher in the small panel, but basically the same, which, that was pretty promising. And then we looked at the on-target depth, and this we put in a relatively small amount of DNA. So we're not going to extreme depths of these particular comparison samples. What we saw was that both the mean and the peak depths for our panel that we spent several months or multiple months optimizing, and this directly designed panel from Twist gave very comparable results. So if you sort of sort all the different intervals that we're looking at in this pretty complex set of regions that are hard to work with, we got very similar profiles for both, which I was pretty surprised by. There were actually some regions where the Twist probes actually did a little bit better than what we had been able to do, and in these areas that have sort of known secondary structure and sort of GC balance issues. And so, whatever automated algorithm they were using actually worked a little bit better than what we could do by hand. So overall, I was pretty pleased with how well this worked. So we did 2 panels using just this, very straight up, here's an interval file, design us a panel. The panel that showed up from Twist 3 weeks later worked pretty much as well, or if not a little bit better than what we had done that required a couple months to order, to run several tests, to optimize, to rebalance, to do X and Y and Z. We got the same results in much less time, and we didn't have to optimize anything. So that was actually really good proof of concept in my view of this, which I did not expect it to work that well. We as a company at TwinStrand we have panels and assays that are sort of set up for very specific applications. But one thing a lot of our customers are wanting is customizable solutions where we simply have an assay and say, we have all the chemistry and software, and they generate probes and drop them in for their own specific application, like a dog canine panel, for example, which is not something we've worked with. And and now it's very easy for us to recommend that this is a very quick, efficient turnaround that's directly compatible with what we do. So I'll stop there and take questions.